Hey everyone, welcome back to the Biochemistry SI Program YouTube channel. Today, we will discuss post-transcriptional processes. Eukaryotic genes consist of coding sequences of nucleotides called exons and non-coding sequences called introns. The product of transcription, called the primary transcript, is formed with a 5' cap and a 3' poly A tail. The 5' cap consists of a modified guanine nucleotide added to the 5' end of the mRNA. It protects the mRNA from degradation by exonucleases. It also facilitates the export of mRNA from the nucleus to the cytoplasm. It is also involved in the initiation of translation by helping the ribosome bind to the mRNA. The 3' poly A tail is a long sequence of adenine nucleotides added to the end of the mRNA and increases the mRNA stability in the cytoplasm. It also assists in the termination of transcription. The intron sequences are then removed by the process of RNA splicing. The intron-exon junctions are recognized by small nuclear ribonucleoproteins called snRNPs, fondly known as SNRPs. These SNRPs then cluster together, forming a larger complex called a spliceosome, which carries out the removal of the introns. You should know that in general, the spliceosome is the nuclear complex responsible for removing intron sequences and ligation of exon sequences 5' to 3' to generate an mRNA. Splicing itself involves the cleavage of the 5' end of the intron, forming a looped structure called a lariat, joining the two exons together and releasing the intron. Remember, introns are released and exons stay. You can remember this by the phrase, exons are expressed. One advantage of the splicing process is that a single primary transcript can be spliced into different messenger RNAs by the inclusion of different sets of exons, also known as alternative splicing. Alternative splicing is important because it allows a single gene to produce multiple proteins with different functions. This process increases the diversity of proteins that can be synthesized from a single gene, which is essential for the complexity of living organisms. By including or excluding certain exons during the splicing process, cells can generate a variety of protein isoforms that have different roles in the cell, contributing to the regulation of gene expression and the adaptability of an organism to its environment. Be sure to not confuse alternative splicing and polycistrionic mRNA, as they sound relatively familiar. Alternative splicing occurs in eukaryotes, where genes are typically monocistrionic, or one gene codes for one protein. Psst. Hey, it's Isabel from editing. Just so you know, we also have a video on alternative splicing on our YouTube channel. If you want to learn more, I'd definitely suggest checking it out. Polycistrionic mRNA is transcribed from operons, which are clusters of genes under the control of a single promoter. I hope you enjoyed the video. We look forward to seeing you in the next one.